Thanks, Francis. It's good to be back. Can everybody hear me? Okay. I have spoken about this before, and what I thought I'd do was I'm going to give a very brief introduction to our SIFA work, which some of you will have heard before, and then I'll go on to give. There are 3 different presentations I've given at conferences over the last year, and so I've put them together into a story that I'll be telling today. So. My title is Drought and its Aftermath in Western Canadian Aspen Forests. And although this presentation is by me, I always like to start with acknowledgements in case I run out of time, because the work that I'm going to be talking about today really wouldn't have been possible without a lot of collaboration and especially summer student field assistance. So I won't go through the list here, but some of you will see your names listed here. The work I'll be talking about covers about the 14-year period, and these are the funding sources that, among others, that have supported this work. Well, many of you have heard, as you've probably heard, certainly drought-induced forest decline has become an emerging global change issue, especially over the past few years. And my colleague Craig Allen with the U.S. Geological Survey, really I think his review paper in 2010 that I was involved in, sort of put it on the front burner of awareness, I think, among researchers in forestry. And since his publication, if you don't know about it, he basically is documenting something like 88 different examples of recent forest decline related to drought on all the major forested continents around the world. It was more of an overview, not a lot of proof, you might say, but other than the fact that the number of publications has been increasing on this topic and the attention. But since then, there have been a number of analyses, and I've listed some of the publications that have shown up recently, and including those relating directly to our Canadian boreal forest. And you might have heard of the Ma et al. And Peg et al. Papers. And then Han Chen's group has also been working on this topic as well. And the focus of the work is trembling aspen, which probably doesn't need much of an introduction, the most widespread tree in North America. I wanted to draw your attention to this recent review or overview of aspen decline in Western North America. It was led by my colleague Jim Worrell with the U.S. Forest Service. But there was, Andreas Hamann was involved and Laura Gray, myself and Mike McHaley. So part of this was providing a review or overview of recent episodes of aspen mortality and their causes. And then another, Andreas, where his work came in terms of looking at the future GCM scenarios and applying his climate envelope models. Basically, it's not really looking like good news for aspen, especially as you go into the U.S. It's just given the trend toward warming and drying that we're seeing. But I'm not going to talk about this paper per se. It's in Forest Ecology and Management, if you're interested. But before I start sort of the new material, I want to just give a review of this SIFA study. The acronym SIFA stands for Climate Impacts on Productivity and Health of Aspen. And it includes a network of plots that we established in the year 2000. And we've been monitoring most of these plots annually up to 2012. One thing that's unusual about this study is that about half of the plots are located outside of the commercial forest in the Aspen Parkland zone down here. And here in Edmonton, we would be what I would consider this area, at least above the river valley, to be a parkland zone. So that gives you an idea. It's a transition between forest and prairie. Another distinctive feature of the study is it's a sort of a multi-scale or hierarchical design aimed at enabling us to scale up from things like tree rings, plot-based measurements, to make inferences of what's happening over larger areas. And we do this through applying simple biomass equations like height, diameter, measuring height and diameter and then inferring biomass changes from that. And then we do annual monitoring of tree health as well as the incidence of insects and diseases at the tree level. So one of the things that came out of this work very early on was just the difference between aspen growing in the parkland, which is a dry climate, compared to the boreal. And we selected these plots, we matched them in terms of age. So it turned out they're exactly the same average age, 60 years when the plots were established. Exactly the same mean stem diameter. What was different was that the trees in the parkland are stunted. They have an odd growth form and lower density, lower biomass by about 37%. The main difference between these zones climatically is mean annual temperatures about 2 degrees C warmer in the parkland. Precipitation's about the same, but it translates into drier conditions in the parkland zone. And then one of the tools that we've been using is tree ring analysis. And one of the ways that we can infer the causes of reduced growth or narrow rings is through these distinctive rings called white tree rings, we call them, or pale colored. They're low density. And we had a project here in the department, a master's student, Miranda Hart, had a study with artificial defoliation of young aspen, and we were able to actually create these white rings experimentally. So it's a useful tool for retrospectively looking at where insects have been defoliating the trees. And that photo there is just shows if you haven't lived through a forest tent caterpillar outbreak, it can be pretty dramatic. You know, basically they go after pretty much all the leaves are gone if it's a severe outbreak. And then we took our first set of tree ring measurements to look at aspen growth over the last previous 50 years, and to understand what's driving that growth variation. So this is just the average growth response over all of these plots. And you can see, one thing you can see right away is these dramatic oscillations, sort of almost decadal level, periods of very good growth, followed by collapses for many years. And from regression modeling, we were able to show that it's primarily drought in combination with insect defoliation that's driving that variation. Growing degree days plays a role as well, a secondary factor, so warmer springs will cause increases in growth as well. But so you can get a 50% drop in growth, for example, from the late 70s to early 80s, here to here, even across very large areas in growth. So it is significant, potentially significant to things like fiber supply and so on. And then to assess drought, I've spoken about this before, but it's the climate moisture index that I know some of you are using here. It's just a simple index that we developed to account for the fact that temperature leads to a more rapid evapotranspiration. So we calculate P minus PET. P is annual precipitation. PET is conceptually the potential rate of water vapor loss from a well-vegetated landscape when soil moisture is not limiting. And it's designed to be simple, so all you need is T max, T min, and precipitation to calculate it. There are better models out there, but we don't have the data for remote boreal regions over long time periods. And then, so this is just an example from our siphon network where we've calculated this moisture index from weather station data going back to 1950. And you can see right away there's this very exceptional drought in 2001, 2002, especially in the parkland. And then so that's temporally, and then spatially we can use interpolation methods like biosim or anusplin, prism. There's a number of different methods that people use, ways of inferring the climate at forests in between climate stations, essentially. So these negative values refer to dry conditions. You can see that red. So in this 2001, 2002, we had essentially a northward movement of prairie-like climates into the boreal forest, or into the southern boreal forest, which led to these impacts that I've talked about previously, 30% drop in regional aspen growth, massive dieback and mortality that was documented in the Global Change Biology paper led by my colleague Mike McKillian, where I think we estimated 45 million tons of dead aspen in this study area between Edmonton and Saskatoon. But certainly other species. If you've lived in Edmonton for any length of time, you'll know the impact that the drought, that drought and subsequent droughts have had on the urban forest around here, especially birch, a lot of dead birch and even white spruce mortality. Although it's certainly a negative impact for a lot of people, for us it provided an opportunity, having established this network, to look at what happens when you have a drought. How long, we've now had 10 years since the end of this drought to look at the impacts. So this brings me to the new stuff, and I'm going to be, the overall theme is about processes affecting multi-year lags or multi-year impacts on aspen forests. I'll start with looking at growth lags, and I'll be focusing on the role of soil moisture as a factor affecting growth lags. Secondly will be drought impacts on tree mortality patterns following 10 years of measurements. And then the question, what happens to trees after they die, how fast do they fall down? So I'll be looking at some of the factors affecting fall down rates of snags. And I'm drawing, because I was asked to come at short notice, I'm drawing very heavily on previous presentations, these three that are shown here, that I've given over the last year. So the first one is relating to growth. And this is a presentation I gave at this tree ring conference in Tucson, Arizona, last May. And it's work with my colleagues Alan Barr with Environment Canada and Andy Black at University of British Columbia. And it's about applying a simple soil moisture model for looking at drought effects on aspen growth in Western Canada. So just bringing you back again, we have this climate moisture index tool, which has been very useful for a variety of applications. It's been a useful tool, but one of its disadvantages is it's really a fairly indirect measure of actual soil moisture, which is one of the main factors that is actually driving drought impacts in forests. So felt that we needed a more realistic indicator of soil moisture, and especially if you go to eastern Canada or the coast, the CMI doesn't work very well in those areas because it's overwhelmed by winter precipitation. So we're interested in seasonal drought effects and in multi-year carryover of soil moisture from one year to the next. So to do this, I developed a simple soil moisture index. It's certainly, again, it's not the best model, but it's a simple model. And all it needs is the same inputs as the CMI, Tmax, Tmin, and precip, because that's all we have if we want to map over large areas and long time periods. So it's a single layer soil water bucket model. It can be run on a daily or monthly time step. And calculate the PET the same way as the CMI, but then we actually we estimate what the actual water use of the forest is, actual evapotranspiration, as a function of PET and soil moisture. And this graph just shows one of the methods we use to, as the soil dries out below here, we start, the dry soil start forcing down evapotranspiration compared to what it could have been if there'd been adequate moisture. And then it's run on a daily time step with this, it has runoff as well. And so here's, one of the reasons I did this was just there was a really good opportunity. You may have heard of the Boreas study that set up these towers. One of them is in Prince Albert National Park. It's an aspen forest, and we've got long-term measurements through these studies, BERMS and FluxNet Canada. There's been a tower measurements measuring soil moisture, among many other things, with TDR probes going down to 1.2 meters. And so we have this sort of 10-year data set that covers this severe 2001-2002 drought that I mentioned. It has some other droughts as well, and then followed by this quite wet period that's actually persisted off and on ever since in that area, as well as the evapotranspiration measurements from eddy covariance. So on the left you'll see what I did here was to tune the model into, all I have is two parameters, but there's water holding capacity in the rooting zone and critical water holding capacity. So those were tuned, admittedly, but nevertheless it gives a good fit to the actual data. Surprisingly, it performs surprisingly well given how simple it is. And then it also does a good job of emulating annual evapotranspiration as measured at the tower. So in an average year, you're running about 350 millimeters or 400 millimeters, say, of water loss over the year. It's not capturing the seasonal factors very well because the model doesn't specifically model leaf out and leaf fall. So it's treating it's simplistic in that way, but it performs reasonably well for this application. And then I won't get into details, but we use unconventional methods in our tree ring work because since we're looking at climate change, we're interested in decadal variation. We don't like detrending methods that take out all the wiggles in the signal. So we use this, in this case, quadratic detrending with basal area increment, which is a closer proxy to biomass. And this is just explained in the paper, so I won't get into detail here. But so again, just bringing you back to the earlier results, we used this climate moisture index to as an indicator of drought. And the problem with the CMI is that in the regression, we have multi-year lag effects in there. So up to 5 years prior to the current year seem to be moisture balance between precipitation and potential evapotranspiration seems to be affecting trees for up to 5 years based on that model. We can do the same analysis with the soil moisture index, which is a closer, more of a estimated soil moisture. And what we see there is we drop out all the lag terms. So now we're only dealing with the current year's soil moisture as being significant in this regression. Now, the problem here is that, as I've mentioned earlier, we have a seriously confounding influence of defoliation. Those purple lines indicate years of severe defoliation. So that could be smearing out some of the climate effects. So it poses some problems. The one way, fortunately, Ross Weiner used to be here in the department, and Ian Nalder did some tree ring sampling up around Fort Smith a few years ago. And that's an area that, until recently, is too far north to have had severe defoliation. So we use this as sort of a test bench to run this model in an area that doesn't have significant insect impacts. And these are older stands that were all close to the Fort Smith Airport, which has a nice climate record going back to 1915. So it even had a longer time period to work with. Applied the same methods as in the previous study, but just with these stands at Fort Smith. And what you see here, here's an area that hasn't had a lot of defoliation. We're still see this green line? This is going back to 1915. We got these dramatic oscillations in growth going up and down like this, especially in the 40s and the early 50s. Well, it turns out that if we run this, either one of those indices, climate moisture index or soil moisture index, suggests that drought is really the main driver of this. But again, with the CMI, we have to include these lag terms up to 4 years previous. With the soil moisture index, we lose them all, except for the previous year. So that's suggesting there is something else going on there. Things like carbohydrate carryover. There's other mechanisms happening that are leading to delays of aspen responses to drought. But it's a much cleaner model, you might say, because it only has one lag term. And then when we, if we plot soil moisture index for the current and previous summer on the x-axis and growth index on the y-axis, we see this very strong relationship, which convinces me that moisture is really driving this. And the other thing I'll just briefly mention, growth has a high serial autocorrelation, aspen growth, R equals 0.81. The soil moisture index, it turns out the CMI is almost like white noise, but when you run the soil moisture index, it is also autocorrelated and explains a lot of the autocorrelation in growth that we're seeing in the aspen here. And so it's interesting because this sort of thing has never been reported from other systems. They talk about soil moisture memory more in the range of months in the meteorological literature, but we think this is sort of a special case here. We have a region that has a very long period of frozen soils where the moisture conditions are preserved for 6 months every year. We've got high water holding capacity, aspens deeply rooted, and good soils, low annual precipitation. So we think those three things combined result in a much greater memory of the system to precipitation events that might have happened in previous years. And there's a recent analysis by Coops et al. In a combination of remote sensing and modeling where they attempted to map the water holding capacity of soils in forested areas around North America. And you can see these blue areas have high water holding capacity, and our aspen forests correspond to those. So there'll be many other vegetation types that may not show this because of the poorer soils or shallowly rooted vegetation. So I guess I'm going to say that I'm going to hold the conclusions until the end for each one of these sections. So I'll get back to this again. But now, in the interest of time, I'll move on to the next part, which is looking now at mortality, and specifically, 10-year mortality from our updated CIFER results. I presented this at the Ecological Society of America conference in Minneapolis last August. It was a session that was organized by Han Chen at Lakehead University. Some of you, he was here speaking last year, so some of you may have met him. And really, our work on mortality feeds into this bigger question that I alluded to at the beginning, this science question for the boreal forest. Is climate change leading to increases in tree mortality? And I'm not going to answer that question here, but what I will say is that it's not as simple a question as you might think. There are major challenges scaling up our plot-based, tree-based measurements to large areas. I've alluded to the forest tent caterpillar, but there's these stochastic, cyclical impacts of insects, which have big impacts on growth. And how do we distinguish between changes in tree mortality induced by normal stand dynamics, self-thinning, things like that we learn about in forestry, tree aging, inter-tree competition, versus climate change. It's getting drier, it's getting warmer. And there are papers that sort of emphasize different sides of this. I think the ones on the bottom maybe are getting more attention, but certainly there's Thorpe and Daniels. Jan Zhank did a PhD here recently, highlighting the role of competition and stand dynamics. So it's not an easy question to answer. Again, just reminding you about this 2001-2002 drought, which is where we also were looking at mortality impacts. And I'd like to now say that we started this in 2000. It's hard to keep a project running over this amount of time, and one way we've managed to keep it going this long is through collaborations. And one of the main ones is with Alberta Environment ESRD. I think it was called SRD before. Our colleagues Mike Underschultz and others have been monitoring our CIFA plots over the last 3 or 4 years in Alberta, which without that sort of help, we would probably have had to shut down the study earlier. So I'd like to now sort of update these mortality results. And as you might have heard me say earlier, the drought led to increases in tree mortality at the regional scale. But there are different ways of expressing this. You can express it on stem number. And what we see there is very similar responses in the boreal and the parkland. We got a approximate doubling of tree mortality that's persisted after 10 years. And only recently there are hints that it's coming down again. So it's been up for a long time. But we get a different story if we express it based on biomass, calculate which trees are actually dying. Now we see the parkland has about twice as high a mortality as the boreal when we do it based on biomass instead of stem number. And so this suggests that drought is having differential impacts according to relative tree size. Drought may be killing larger trees. So to illustrate this, we looked at the boreal forest sites, the sites in the boreal, and this time we're sort of drilling down to look at which size classes are actually dying over that 10-year period. And the boreal forest areas they did have drought, but not nearly as severe as the parkland. So what we see here is that, if you're a really small tree, less than 0.3 of the average, almost 90% of those are dead after 10 years, which is what you'd expect from self-thinning Larger trees, hardly ever dying. Now we go to the parkland. We're seeing a shift, a shift in the mortality pattern. The medium and large trees are now starting to die significantly, and in some sites dramatically so. And the small ones are actually dying at a slower rate, perhaps because the canopy is opening up and they're able to survive a little bit more. And so there's this concept, I call this the RB, relative RB is the tree biomass compared to the average tree biomass in the stand, just to remind you. And then we can also express this, instead of just dividing parkland boreal, we can look more specifically at the drought severity in these different sites during that severe drought year. And what we see is on the right there, these are some sites that didn't get any drought in the boreal forest. The average tree that died was about a third of the average in that stand. And as you get to areas with more and more drought severity, we're starting to see mortality being spread more evenly across size classes. So larger trees are dying following drought. And I'll wrap up this one too at the end. So now I'm going to move to the next part. I know this is a bit of a whirlwind so now I'm going to talk about factors affecting fall down rates. Okay, once the trees die, how fast do they fall down, and what causes it? And this is a presentation I gave at the Western International Forest Disease Conference in Waterton. This is why I was not at the IBFRA meeting, by the way, because I was organizing an aspen panel at that meeting, aspen decline panel. So I guess you might ask, why is it important? How fast trees fall down? Well, there's a lot of reasons. I guess public safety might be one of them, and bird, habitat. But it's also given that dead trees are a major source of future carbon emissions after they decompose, and it's the fall down and breakage of snags that's the first step of decomposition that brings them down to the forest floor where it's moister and they're in contact with soil decomposers. So the rate of fall down we believe is important to the carbon cycle. And so we also then used our CIFER results to look at this as well. And these are the snags. This is a 5-year repeat photography at one of our sites where 5 years later, this was actually a healthy stand when we established it. This is what it looked like in 2008. And these are, you can see the snapping. These are the tops of the trees snap over. Very, very rarely do aspen actually blow over while alive, maybe because of their root systems. But so we did an analysis of snag fall from this annual plot-based assessments. We had 1,010 trees that died that we could look at, and we developed a method of estimating the biomass contained in those top parts that fall down. And I won't get into detail about that, but it's a fairly complicated calculation, but anyway. So again, as I mentioned for the living trees, we're also looking at results according to stem number or according to biomass. Most previous snagfall studies have looked according to stem number. And what we see is, first thing is that the dead aspen fall down very quickly. After 5 years or so, less than half of them are standing and not snapped. These gray areas, these hatched areas represent the trees that are snapped. So within the first few years, there's a lot of trees snapping halfway up the tree. And then after 10 years, there's not a whole lot left standing. We can also express it in terms of stem mass. And if you pool all the sites together, you get this nice negative exponential, which is good for modeling. And what we see is about a 1-year lag. The first year after death, hardly any trees ever fall unless there's a tornado. But then they start falling at about 19% of the biomass falls down every year on average. So again, after 10 years, there's not much left standing. But this sort of is glossing over a lot of high-level variation from one site to the next. So you see sites like Fort Nelson up in northern BC, the fall down is very slow there. The tower site where we did our soil moisture work is pretty much on average representative of what's happening regionally. And then we have sites that are falling down very quickly, like Porcupine Hills in Manitoba. And then you can see here, these different numbers represent the K rate, the percent fall down per year. So you can see the blue numbers in northern Alberta, relatively slower fall down rates. Redder areas, higher fall down rates in the parklands, especially in the eastern part of our study area. So we did a regression analysis to look at the factors affecting fall down, including climate variables and biotic variables. There was a lot of analysis done. And what I'm just going to show you the most important factor that we identified was the incidence of Phylinus tremulae, which is a common decay fungus that weakens the trees. It colonizes the trees as they get older, and we see a good relationship there. Stand age was also important, or windiness, site windiness was also important, a secondary factor as well. So, not surprisingly then, the incidence of Phylinus, when we established the study, shows a fairly similar pattern to what we saw in terms of fall down rates. So northern Alberta, again, very low incidence of Phylinus at the start of our study, less than 5% at most of our sites, 0 at some of our northern sites. And in general, the parkland had much more incidence of Phylinus. And then here I'm going to go 10 years forward. 2012, the numbers have gone up. Part of that's just natural aging of the stands, but illustrates it's getting higher in the parkland especially. So then you might ask, okay, well, if Phylinus is driving, a main factor driving fall down, what drives Phylinus? And well, one thing we see is stand age is important. Younger stands hardly ever have much Phylinus, at least visible symptoms, and as the trees get beyond about 80 years old, they start showing more. Did a lot of analyses. Turned out that mean annual temperature was the best secondary predictor besides stand age, and separates the points pretty well. So these warmer sites in the parkland tend to have earlier, we might say earlier onset of decay. Statistics might be a little bit shaky there, but I think you can see there's some indication that the warmer temperatures lead to earlier onset of decay, which it fits with some of the other literature that's out there. So what does that mean for climate change? We've already seen about a 2 degrees Celsius warming over the last 100 years. This is based on biosim reconstructions of all our SIFA sites. So that our sites in the boreal forest are now as warm as they were in the parkland 100 years ago. And of course, we don't have long-term data on incidence of Phylinus, but I think it's possible that Phylinus is increasing due to warming. And so for this conference presentation, I just put together this simple conceptual model, sort of illustrating how basically it was a meeting of pathologists. So I wanted to highlight the importance of fungi in climate change studies. And so of course on the left, climate change leading to warming, increased drought. If the warming is increasing the Phylinus and the drought is increasing aspen mortality, then when you put the two together, you end up with more decay, more rapid fall down, and potentially greater carbon losses, partly mediated by fungi. So now I'm at the summary and conclusion slides for the 3 different components. So I'll now just go through them to remind you where we were before. So in that first study with growth lags on the soil moisture model, we developed that using 9 years of measurements at a flux tower site. And it turned out to be a useful tool for tree ring analysis that suggested that multi-year growth lags are mainly a consequence of soil moisture memory. Maybe not mainly a consequence, but at least partly. And I guess speaking to dendro people, suggesting that tree ring studies might benefit from applying actual soil moisture models as an alternative to the methods they traditionally use, monthly precipitation response functions, which is I think a coarser way of looking at drought. Oh, and I'll just mention there's a paper out, and I'll be glad to send it to you if you're interested in more details. In terms of tree mortality patterns, certainly we've seen a 10-year sustained increase in tree mortality. I didn't mention some of the mechanisms. One of them might be the increases in wood-boring insects, for example. So insects and diseases may have played a role in amplifying drought effects, which I didn't get into today. And I'm suggesting that instead of using just percent stems dying, as some of the early studies tend to do, we should be looking at the size distribution of these dying trees, which can help us to separate things like negative effects of drought and versus stand dynamics. And I know that Yang Luo has done his PhD and has seen this in his work as well, but there is a suggestion that in some regions, CO2 fertilization and longer growing season is actually leading to more rapid growth of these forests, which accelerates the self-thinning process. So if you're just simply plot mortality by stem number and so on, an upward trend, it really doesn't prove anything. It doesn't tell you anything about climate change. So you really need to get into the mechanisms, put your understanding of stand dynamics into these analyses of climate effects. And then the third part is on fall down of drought-killed snags. Our rates of fall down were more rapid than what some have reported earlier. But the variation we're seeing is at least statistically related to the incidence of Phalinus. It might be serving as a proxy for other factors like other fungi. But Phalinus appears to be positively related to mean temperature. And given that we've seen warming over the last 100 years, it may be that some of these fungi are actually increasing out there, although we don't have a lot of records. And certainly, based on all three of the talks, I think the projected warming and drying certainly poses a high future risk for both aspen decline and associated carbon losses in the future. So that's the end of my talk, and I'll be glad to answer any questions.